Well, good morning, everyone. Nice Friday morning. Let's shift over to the computer and get things going. Good morning, everyone downtown. Nice to see you today. Looks like a few new people, so remember, if you have a question, and here as well, push the little button in front of you. And if you don't want to be on camera, just put a notebook in front of your face, I guess, and let it pick up your audio so we have a record of the questions and people will understand the response when they're just watching the streaming video. I hope everyone has had a chance to navigate around the WebCT site a couple of times, and hopefully those that are doing the online section have had a chance to look at Wednesday's lecture, the streaming video. And today we will jump to the scheduled lecture, Lecture 2. And what I'm going to do now is go to the WebCT and show you the previous semester's in-class recorded streaming video lectures that contain the Lecture 1. I sent an email, but was kind of clicking around today to find it. So let's switch over to that. I forgot to put myself in the pip. There we are. So under the lectures on the streaming video, the class lectures for fall 2003, and we know that class is misspelled on the one for this semester, but we'll just leave it that way. So the second half of August 25th, and then just about the second half, you'll see we're having some technical difficulties on the 27th. On those two, you will find the first lecture, and then today's lecture isn't present there. If you want to get a jump on the next one on the weekend, the August 29 picks up with Lecture 3. So it's all up to you for those individuals doing just the online course. Keep pace and keep your own pace. So you're free to do all that. So let's switch over then to today's lecture, Lecture 2 on meiosis. It's essential to understand this. The Lecture 1 is really just an overview of the entire developmental period, and just get you geared up to have an appreciation for where we're going to go. Basically from meiotic activity for gametogenesis to birth is where we will go with a varying degree of detail as we proceed. And of course, since this really is a clinically oriented human embryology course, we will have case studies. We'll look at anomalies, developmental anomalies, and a variety of things like that. There will also be a lot of slides, so I stated that this will be gross anatomical and micro, so macroanatomy and microanatomy, the histology for the latter. And we will try to develop our skills in studying gross anatomy, albeit at the embryonic fetal stages, and histology, the microanatomy. So we'll add that in case you take another course. Let me set my pointer here so it stays on all the time. So obviously we need to talk about meiosis in order to appreciate what occurs during the gametogenesis. Obviously this should all be reviewed. Let's see. We lost the downtown people up front there again. But guess you all don't need to be staring at each other. We'll just proceed. We start off with the overview that obviously this is the specialized cell division of the egg and the sperms, so therefore we proceed from this lecture to gametogenesis, the spermatogenesis, and oogenesis. And this is going to produce more cells, but they are genetically different from each other. So the cell of origin will have really a different genetic makeup, not the sequence of DNA so much. To some degree there might be changes in that, but really the allelic assembly on any one chromosome, as we will see. And that's our effort today, is to review the transmission of genetic information at this level of examination of the chromosomes. So we want to remind ourselves of the difference between mitosis and meiosis. In mitosis, well, let's back up. For any organism to survive for any period of time, multicellular organism, let's say, you've got to replace cells. cells die. They have a limited lifetime. Is that true of all cells in a multicellular organism? No, we have to have reserve cells. So there's a variety of different types of reserve cells that actually reserve mitotic capability, as well as being able to send the daughter cell into meiotic division for what we're discussing here. So the production of a new cell is not at the expense of the reserve cell, other than energy and proteins and all the essential molecules for producing a new cell. We have a reserve cell that divides and produces two identical daughter cells, is the standard thing that we all recall, but indeed it doesn't. There's an identical daughter cell, but mommy stays there as the reserve cell. Doesn't go away. If it went away, we'd lose our reserve cells. So keep that in mind. So the reserve cells for gametogenesis do persist. Do they persist into adulthood and senescence in both males and females, the reserve cells? Start with males. Do males have the capacity to produce sperm from reserve cells even into their 7th and 8th and 9th decades? Yes. How about females? No, the reserve cells disappear during development. At birth, there are a certain number of primary follicles, and there's more than enough to have all the children anyone might want to have. So there's a nice reserve, if you want to borrow that same term, of these egg-producing cells. So when we talk about the female gametogenesis, the oogenesis, we will see that we discuss it in the context of what's going on in the embryo and fetus. Then when we talk about the ovarian cycle, we'll refer to, obviously, to the adult ovary where there are no more reserve cells. At least this is the standing knowledge as it is today. So other than that idea of appreciating that there's reserve cells and they produce a new cell that with mitosis, say for the gut lining, is going to differentiate to a terminal differentiated state and produce its function and then die and just go away. Here we're talking about a reserve cell producing another cell with a mitosis, and then that cell undergoes the meiotic division to produce the gametes. In coordination with that, of course, we have to think about the chromosome number and what's happening. So how many chromosome pairs are there in a somatic cell? Okay. Little brain fart there. I can't remember myself. But nobody shouted it out, so it seems everybody's a little reserved about it. Well, we'll work that as we go along. Well, there's 23 pairs, right? 46 chromosomes. And what we're going to do with meiosis, obviously, is end up with half of those. So there's, on the maternal side, the maternal contribution of half of the genome, if you will, and the paternal side, the contribution of half of the genome. So nothing new for any of us yet. But in the process of getting to that point, there's a random assortment of traits of the alleles on the chromosomes. So this slide summarizes this process. We have to produce a new cell from a reserve cell, and it's going to undergo a process producing the gametes. And this is just a visual starter for us. We'll go into the details starting with this slide. So the panels that we'll be looking at will have a histologically stained cell, and we will follow it through the illustrative and descriptive parts in the lecture and reflect on what's happening with the DNA as we proceed. So to start with the histological specimen, we see the dark brown stained structure, which obviously is the nucleus. So the nuclear proteins are picking up this basic dye and staining blue. And then cytoplasmic elements are picking up the acidic stain and staining red in this case. There's a lot of different types of stains. This one I'm assuming is hematoxin and eosin that produces this type of an appearance. So obviously the nuclear membrane must be intact. It's enclosing the nucleus. And what we know is happening here at interphase is that the cell is basically living a phase of its life. In terminally differentiated cells, the interface is where it stays, but it doesn't go through the G1, S, and G2. It's locked in the interface, and it's going to live its life and die. So it's the reserve cells that have, or their daughter cells that reserve some mitotic capability. For example, immune cells, like the B cells, reserve mitotic capability. Our memory cells can be triggered by a second visit by an antigen and undergo mitosis again. So not every daughter cell is going to lose its mitotic capability. But obviously in meiosis, that will occur once meiosis starts. Oh no, there are divisions in meiosis. What am I thinking of? So it's reserved at least to go into the second division phase. So it's during this phase of the cell cycle that the replication of the DNA is going to occur. We have to do this because we're going to produce the cells that are going to undergo the meiosis. So we have to have a mitotic division that feeds into the meiotic cell division. So there's the three cycles, the subphases, G1, S, and G2. The mitosis is somatic cell division, and then the meiosis, as we know. So before we push on, we need to keep in mind that there's terminology for nuclear division, and there's terminology for cellular division, or cytoplasmic division, if you will. All right, so the nuclear division is pretty much what we're going to focus on. The cytokinesis is an entirely separate and very dynamic series of events in a cell where we replicate all of the subcellular organelles, the cytoplasmic content, or at least we don't really replicate it, but we produce enough so that we can, give two separate cells with all the appropriate machinery after the mitotic division and the cytokinesis. So what I want you to think about is how the endoplasmic reticulum comes and goes. So what happens to the ER, to all the subcellular organelles when a cell divides. They're very dynamic structures. They're not static. So endoplasmic reticulum can disappear and then be reassembled. And we know that membranes, intracellular membranes, can do this because with the mitosis, and meiosis, the actual nuclear division, we're not really dividing the nucleus. We're shedding everything except for the DNA. So where do all those proteins go? Where does all the membrane proteins and the phospholipids of the nuclear envelope, that doublet of lipid bilayers, where do they go during all of this? And then suddenly they reappear at the end and encase everything in the nucleus again. So we look at this pictorially, throughout our life of studying science, and we sometimes don't stop to really appreciate the marvelous events that are occurring. How does the cell know to take this protein from in the nucleus, move it over to here somewhere, or break it down and destroy it, and reassemble it and put it back in the right place when the cell is going to reform the nucleus? Can it even produce new protein at a later state when it's going to put the nucleus back together? Very likely it can't. So all of those proteins that have to take care of all that stuff, virtually all of them, saying all in science is a very dangerous generalization. But essentially everything we need to go through mitosis or meiosis has got to be produced here and now at interphase and reside somewhere in the nucleus, get to where it's supposed to be when it's supposed to be there, do what it's supposed to do when it gets there, stop what it's doing when it's not supposed to do it anymore, and then disappear or go into a storage site for some other type of utilization. So all of this is occurring in G1, S, and G2 in the cytoplasm as well as within the nucleus. So if we are going to have the DNA and the centrosomes replicated, then we remind ourselves of how the DNA is replicated. So there is this obviously is the double helix that is opened up, and the DNA polymerase 3 is elongating the leading strand. The helicase is unwinding the double, the double helix. The single-stranded DNA binding proteins make the templates available for the RNA primase and the DNA polymerase 3. And then the RNA primase makes the primer, and then the DNA polymerase 3 elongates the lagging strand. So we're envisioning this exact replication of the DNA. I bring this in not so much to force you to know all of this. Again, it's a little peek at the grandeur of what is occurring here. And it also reminds us that we start with one strand of DNA for each chromosome. And again, it's this definition of chromosome that we're working on. What is a chromosome, and when is it this kind and that kind of a chromosome? So that we can keep track of when we've got the events occurring that we're going to focus on. So this is what we think of when we think of DNA, the parent DNA, the double helix, and it is all coiled. Well, no, it's in the functioning cell. It's all uncoiled to whatever degree to expose the genes that are going to be expressed, right? So that's the way it is here in the nucleus of the interphase cell. So we've got euchromatin and heterochromatin. The euchromatin is the lighter colored stuff. It's unwound, available for enzymes to come in and express genes. And we would have to do all of that in interphase before we got to the DNA replication part. If there's any proteins we need to produce to prepare ourselves for what's going to be coming ahead, we've got to do it before we start replicating the DNA. So we've got gene expression. We'll shut that down. Then we shift over to DNA replication. This occurs, and from the original parent DNA of the two strands, the antiparallel strands, sense and antisense, we produce two sets of the exact same DNA. What's the term for the feature where we see, what's the blue and the red representing? What's the term that we use for that? Is that semi-conservative? So one strand is still the parent strand. The blue strand, the red one, is the brand new DNA that we just made. So just a bit of review. I'm not going to chase you down on this. But what's important is that this is going to become a chromatid, and this is going to become a chromatid. And when those two chromatids are together, we're going to call them a chromosome. So this is a chromosome, this is a chromatid, this is a chromatid. Later, after we finish replicating that whole strand of DNA, we hold them together, and each one is called a chromatid. It's still double-stranded DNA called a chromatid because it has one strand of parent and one strand of brand new DNA. And we will hold them together. That collective structure of the two semi-conserved pieces of DNA is going to be called collectively a chromosome. So we're going to have an unduplicated chromosome and a duplicated chromosome. We'll see this as we proceed, but I wanted to take this illustration to show us how one chromosome ends up becoming one chromosome, but we've got two strands of double helix DNA. So the approach that I have taken is that it's somewhat of a Socratic approach, I guess. As material is covered, sample questions are inserted into the lectures for you to answer as an exercise just for yourself in preparing for the exams. That way when you see exam questions, you're not shocked by the type of questions that are asked. And so I'm trying to alleviate that challenge of waiting for the first exam to figure out how the instructor examines you. So I've inserted those to ease that stress a bit. So this illustration is showing us, of course, the makeup of a coiled chromosome, the paired sister chromatids, excuse me, the two sister chromatids. Let's not use paired yet, right? The two sister chromatids are attached together. Each chromatid of this duplicated chromosome is the double helix DNA. As it's uncoiled, we see that we reach the smallest level of the coil called the nucleosome, where the DNA molecule binds with histones, forming a vast number of these nucleosomes. So there's 2.5 turns on there, and each of those is referred to with their associated proteins is called the nucleosome. So after the nucleosome, it is the fully uncoiled DNA that would be available for proteins to come in, either to begin replication, and again, to think of replication, just forget about all this down here, but this is the state that the DNA has to be in for any type of protein to attach to it and take some action, whether it's producing two sister chromatids in replication, or opening it up so that RNA transcriptase can get in there and we can express a gene. So this would be euchromatin if it were in a cell, and somewhere down here would be the heterochromatin of the more tightly packed DNA that isn't available for gene expression. So again, we're using this illustration to discuss a variety of things, but I take every opportunity I can to help people appreciate the fact that we grow up basically in science saying, oh, here's a chromosome. This is what a chromosome looks like. No, it isn't. In the cells that are functioning in our body, it's one chromosome is just the single double helix of DNA, and for gene expression to occur, some of that has to be exposed at this level so proteins can come in and produce the RNA from it. Which of these, what's the name for these two strands when we start talking about gene transcription? If you think about it, it'll make sense to you. Does that help? The sense and the antisense strands? So which of those strands is transcribed, the sense or the antisense? The antisense strand is transcribed. So that the RNA basically represents the sense sequence. Okay, that's enough of the molecular stuff. I won't be challenging you any type of questions on that. So the nucleosomes actually can be visualized. I know you can't see them, but they can be visualized at the electron microscopic level, so there is data to validate that. So let's follow it all the way back down. We just replicated the DNA as we saw in the previous slide. Now we've got to coil it all up. Even as we took the parent DNA and made the two sister chromatids, somewhere along the way there are proteins holding them together, even though they're all stretched out as nucleosomes or this up here, and then they begin to coil. So that's one of the next things that we have to do. So we have to uncoil all the DNA, replicate it, package it back up, but hold those sister chromatids together through the whole process so that these are basically, in essence, two identical chromosomes, right? But they're not chromosomes. They're two chromatids making up a duplicated chromosome. But ultimately, when they break apart, each chromatid becomes what? An unduplicated chromosome that goes on to another cell. So this slide is showing us a higher magnification of a chromosome made up of two sister chromatids with the centromere in the constricted region holding the two sister chromatids together. And there's always proteins involved in this type of a thing. So this is a a double helix of DNA all coiled up with the nucleosomes, coiled and supercoiled and all of that stuff until it's all condensed, a nice convenient little package for moving around and things like that. So we remind ourselves of that stuff. So every somatic cell in our body contains one of these, right? Is that correct? Every somatic cell in our body contains one of these? No. Every somatic cell in our body contains the chromosome that's produced from this chromatid as a double-stranded DNA. So in mitosis, we ultimately split here to send this to one cell, say it stays in the reserve cell, and this one goes to the daughter cell. See? So we're misled by our pre-college science teachers, aren't we? You never see this in a somatic cell unless it's undergoing mitosis. We're talking about meiosis, I know, but we've got to go through a mitosis before we can proceed into the meiotic division. But even during meiosis, we have to keep in mind, am I talking about a chromosome that is a pair of sister chromatids held together at a centromere? Or am I talking about a duplicated chromosome there? Or am I talking about an unduplicated chromosome that's just the double helix of DNA all coiled up and is not hooked together to its sister chromatid? Therefore, it would be an unduplicated chromosome. Even though it just underwent duplication, once these sister chromatids split, it's now called an unduplicated chromosome. So what's missing from this picture? What was one of the first questions I had asked you? How many pairs of chromosomes do we have in every one of our cells, somatic cells in our body? So what's missing from this picture is the homologous pair chromosome that goes with it. At this stage, somewhere in this dividing cell, there's going to be the homologous chromosome that's like this one and would virtually look the same. Right? So we come back to this slide and we look down here. When we perform an analysis on the cells of an organism where we look at the chromosomes in this fashion, what is that called? When you look at someone's chromosomes and you cut it out with the pictures and you say, well, here's this chromosome, here's this one. Karyotyping, yeah. Okay. So we know that somewhere in here there's chromosome 3, the homologous pairs, but it's actually the duplicated chromosomes that we're looking at. So let's look at a few more simplistic illustrations on this. Again, the question is embedded to orient you to how we might examine you on this material. So one chromosome unduplicated. We have 23 pairs of these in every one of our somatic cells, right? And in our reserve cells, as long as they're in interphase before they've undergone DNA replication. Once we have the DNA replication in the reserve cells, or any cell that, partially differentiated cell that still has some capacity for the nuclear division, the mitosis or the meiosis, we would find a duplicated chromosome. Okay, so one chromosome can become two chromatids stuck together that's still a chromosome. And again, we see this illustration here. In the blue box, we see one chromatid on this duplicated chromosome, and this is one of a homologous pair of chromosomes. So that's important when we go into this discussion of meiosis, because we're going to see different things happen at different stages of the meiotic division. In mitosis, we know that it splits at the centromere, and one chromatid becomes an unduplicated chromosome going to one cell, and the other one goes back to the reserve cell, and from one chromosome we produced two. In meiosis, it's going to be a bit different. At one point, we're going to see that these chromatids don't split, and the homologous pairs are going to go to the opposite poles. And then the next division, those will split at their centromere, and we end up with haploid cells. So therefore, this belabored discussion of the chromosome, how do you define a chromosome? I don't know how you do it. You just got to know what the heck you're talking about and what stage it's working in. So in the meiosis, then, the first division is the reduction division. So the prophase 1 will be the pairing of the homologous chromosomes, the 46 chromosomes, 23 pairs. That will proceed to the metaphase 1, where these pairs will line up at the middle of the cell. So it's pairs of duplicated chromosomes, each homologous chromosome having the sister chromatids stuck together at the centromere. So even though we know the DNA has been replicated, each set of sister chromatids stuck together is one chromosome. So even though we've got all of those in the cell, when those separate, not at the centromere, we've got each of the homologous pairs going to opposite poles, and we get the cytokinesis, then we have reduced the number of chromosomes. We don't have homologous pairs. So the pairs separate to the opposite sides of the cell in the anaphase 1. And we'll go through the illustrations on this as well. And then telophase, you have 2 new cells consisting of 23 chromosomes. These are 23 duplicated chromosomes. Okay, so it's 23 duplicated chromosomes, each of which is made up of 2 sister chromatids stuck together. So those can split at the centromere and produce 2 additional cells, each of which are still haploid, only have 23 chromosomes, and there's not paired. You don't have the homologous pairs, but there is obviously quite a difference in the number of DNA strands you have in that final cell after the second meiotic division. So let's take a look at this through these illustrations then. In prophase, we'll follow it through like an earlier prophase and a later prophase, so we have this greater appreciation for the meiotic events. First, I draw your attention to the cytological slide above. Why do I say it's a cytological slide rather than a histological slide? What's the difference between cytology and histology? Cyto is cell and histo is tissue. Okay? So this cytological sample again shows the proteins in the nucleus staining blue. They're still quite nicely encased by the nuclear envelope, so we haven't ridded ourselves, we haven't eliminated the nuclear envelope at this stage. So everything that happened during interphase to replicate the DNA and produce all these sister chromatids and to sustain our homologous pairs within the cell stayed and occurred within the contained nucleus that was contained by the nuclear envelope. Well, what are you noticing about the cytoplasm? Does that cytoplasm look any different than what we saw in the interphase cell? Does it look like there's little wisps of structures coming through there? What subcellular organelle do you think is staining in that slide, in that sample? Probably cytoskeleton. You've got to have cytoskeleton to undergo any type of movement within a cell, any type of change in structure, right? So any movement within a cell and any maintenance or change in shape of the cell or movement of the cell, which really doesn't occur here, requires the cytoskeleton. So somewhere along the way, we produced a lot of cytoskeletal protein as well as many others. And the cytoskeletal protein tended to pick up the acidic stain. So we are focusing on nuclear division, the meiosis. So in the early prophase, we see the DNA that had been replicated now is going to undergo the coiling and supercoiling and condensation to produce these duplicated chromosomes as we typically see them in a karyotype. And we have the corresponding homologous pairs, blue and red, contained within this cell. As we proceed through prophase, the later prophase, the homologous chromosomes, duplicated chromosomes made up of sister chromatids stuck together at the centromere, they're going to find each other, they're going to pair up, and then we'll proceed, obviously, to certain events before metaphase when they align at the equatorial plane within the cell. So I'll just read the script on here so it draws our attention to what we'll view in the subsequent slides. So the synapsis aligns homologs and chromosomes condense. The homologs are shown in different colors indicating those coming from each parent, and in reality their differences are very small, usually comprising different alleles of some gene. So again, we're thinking that every cell in our body, even reserve cells, has one set of chromosomes from the paternal side and one set from the maternal side. So red's paternal, blue's maternal. So the corresponding questions for you to take a look at. So this synapsis that occurs is really very important because this is when we get the end product of child looking a little bit like mom, a little bit like dad, a little bit like maternal grandpa and Uncle Joe on maternal side, and a bunch of different family members on the other side. There's a shuffling of the alleles, so even though you may receive a maternal chromosome from your mother, some of her father's DNA and mother's DNA, and historically all their ancestors, right, is mixed up within that. It's not identical to what is in her somatic cell DNA, and it's the same of the contribution from the father. So in meiosis, there's a lot of mixing of these alleles. So the alleles are the identical genes, basically. There's slight variations in them, but it's the same gene, or segments of the sister chromatids contain numerous genes, so we're swapping a large number of genes. But the genes that would be on this sister chromatid here in blue will be essentially the same as the ones on there. They're the protein product, the gene product, or protein from any one of those genes still is going to do the same function. It'll do it just a little bit differently because it's a slightly different allele. So in the late prophase or the pro-metaphase, we see that we still have the blue staining, but it kind of looks like the nuclear membrane really isn't containing things. So we're beginning to dissolve the nuclear membrane as we proceed into this. So you can see in this illustration above that the nuclear membrane is beginning to break down a little bit, and the paired homologous chromosomes are kind of sticking to each other in certain areas along their length of the sister chromatids. So during prophase 1, the homologous chromosomes, each with a pair of sister chromatids, so these are homologous duplicated chromosomes, line up to form the tetrad, and then a chiasma forms between the adjacent chromatids of different homologs, and the breakage and rejoining of the chiasma results in the recombinant chromatids. So some of dad's, the paternal DNA ends up on the maternal chromosome that could be contributed to the gamete that produced us. So for a particular chromosome coming from your mother, it will contain DNA alleles from both her mother and her father. And this is a scanning electron micrograph, pretty high magnification, showing these chiasma where the sister chromatids are sticking together, and there's the swapping of the segments of the sister chromatids. So we see it here, A, B, and C representing the genes, the allele, and the uppercase is always dominant, remember, and lowercase is the recessive. So this lets us see that you could receive your mother's maternal chromosome that in all of your mother's somatic cells has the dominant form of the allele, but you might receive it where it will ultimately come to you with it's homozygous dominant, right? It might come to you as heterozygous dominant because it picked up through this process of the recessive allele from the paternal chromosome. Okay, so that takes us through that part so that we appreciate that there's a diversity of new organisms produced that are not exactly like either of the parents or grandparents and so on and so forth. But we end up with still the same human genome. We don't lose genes along the way if everything works well. So it's a very complex process. Then we proceed to metaphase. So the nuclear envelope is obviously gone. So what happened to all those nuclear envelope proteins, nuclear envelope phospholipids? The rough ER and smooth ER are gone. The Golgi apparatus is gone. Where is all that stuff? Ultimately, it's just all of a sudden going to come back. And basically, they're stored in either little spheres of the lipids or actually just dissolved lipids and proteins, and later they'll be reassembled. So these are really smart cells. So the centromeres do not separate. The sister chromatids remain together during anaphase, and the homologs separate. So we see it in the actual cytological samples here. So these are all duplicated chromosomes. Their corresponding homologs are over here. So we can't say that all the red end up on one side and all the blue end up on one side because we just had the synapsis and recombinant events occurring. So the red-blue end up over here and blue-red end up over here. So obviously we're going to proceed to cytokinesis, and then there will be another division that will separate the sister chromatids. What are all these little lines representing in here? Spindle apparatus, which is made up of what? What type of protein makes up spindle apparatus? Microtubule. Right, so they're microtubules. Microtubules are what in what category of subcellular organelles? Cytoskeleton. Any movement within a cell or change in the shape of a cell depends upon cytoskeletal proteins. Okay, microtubules are just one kind. The microtubular proteins. Well, then, obviously, this brings us to the second division. Prophase 2, metaphase 2, anaphase 2, and telophase 2. So we're now talking about 23 chromosomes. What word should be inserted right here between 23 and chromosomes? Whoop. Wait. My picture in a picture is not me. Okay. I don't know what it is. It's a book from somewhere. Weird. The word that should be inserted here is duplicated. 23 duplicated chromosomes. And when we finish this, and we say there's 4 new cells consisting of 23 chromosomes, what word should be inserted there? Unduplicated chromosomes. So an unduplicated chromosome becomes a duplicated chromosome that splits into 2 unduplicated chromosomes. So it's the same. They are condensed. Then they line up at the middle of the equatorial plane of the cell at metaphase. In anaphase, the chromosomes separate at the chromatids. And in telophase 2, there's 4 new cells consisting of the chromosomes each, but only 1 chromatid each. So unduplicated chromosomes. It's not really called a chromatid. Once they separate, they're not chromatids anymore. They are now unduplicated chromosomes. This term should not be here either in the strictest context of what we're discussing. Meiosis is nuclear division. It is not cellular division. Mitosis is nuclear division for somatic cells to produce 2 basically identical daughter cells as far as each one having the paired chromosomes. But we want to remind ourselves that we start with 1 cell and end up with 4. Start with 23 pairs of unduplicated chromosomes, duplicate those chromosomes, we have twice as many, so we now can produce twice as many cells from a typical mitotic cell-like division. So from 1 cell we can produce 4 haploid cells. But these phases that we're talking about are not for cellular cytoplasmic division. They are for the the nuclear division of the entire cell division. And I know sometimes there I said cell division when I meant cytoplasmic division. And so obviously, telophase, we see both cytokinesis, the cytoplasmic portion of overall cellular division, occurring simultaneously with the final part of the nuclear division. So the the unduplicated chromosomes end up at the opposite poles. What's the strict definition of telophase? What's the difference between anaphase and telophase? Anaphase is when they're moving. When they stop, it's telophase. As far as referring just to the movement of the unduplicated haploid number of chromosomes, when they stop moving, that's basically telophase. But many other cellular events begin to emerge as detectable, either visibly detectable or chemically detectable elements within the cell. These are dynamic processes, zillions of metabolic processes occurring, not zillions, but a whole bunch of them, occurring at the same time. So during anaphase, we're not simply having the movement of these unpaired chromosomes toward the opposite poles. There's a lot of enzymatic activity, cytoskeletal activity to produce this cleavage furrow and to assemble the nuclear envelope, putting back all the proteins in there, reassembling the endoplasmic reticulum. Mitochondria will be operating as usual. We would expect that all the glycolytic enzymes and mitochondrial functions to produce the ATP to keep all this stuff going would have been sustained throughout this. So that's one organelle that certainly would have continued without much change, but a tremendous demand placed upon it. So the telophase 1 will end when we begin to see the separation of the cells. So at prophase 2, sorry about that. I just referred to these as the non-duplicated chromosomes, didn't I? They're still duplicated because we're going into the second division now. Back up and dub in the correct words all there within that lecture. Now, when we proceed to the second division, obviously we're going to see the separation of the chromatids producing the unduplicated chromosomes at the opposite poles to complete that. Hopefully that still makes sense to all of you. I'll review that one and see just how much I screwed it up over the weekend. So this ends with a couple of questions, and then obviously in telophase 2 we are producing the cleavage furrows to end up with 4 cells, each of which is going to have the 23 unpaired, unduplicated chromosomes that used to be a sister chromatid. And I'll stop there because we're out of time. And we will go through these last couple slides at the beginning of the lecture on Monday. Have you tried to get on WebCT?
No, not yet.
See, what's more important is whether you're on there.
What are the prerequisites for this class?
Basically Bio 1, Bio 2. Yeah.
So I made it up to Bio 3.
Yeah, you're more than
Just
ready.
making sure. Okay.
Let's see, control
I don't remember this test in Bio 1.
Bio 1 is way too long.
Yeah, it's live. I'm
panel.
trying to remember.
I add to it a bit too. What's your last name?
Alphin, A-L-P-H-I-N. No?
No, you're not in there yet. In the, will you be going to the College of Sciences today, or?
Not today. I can, I guess.
Okay. You can send an email to webct@utsa.edu and ask them when they'll be adding you to this course.
Okay.
Now let me go to Banner ASAP.
So WebCT and then email them?
Yeah, because it's the webct@utsa.edu. They're the ones that take care of that stuff.